Good afternoon, everyone, and welcome to this VIA webinar on the 3 Rs in action, bridging the gap between policy and practice. My name is Emma Lawrence, and I'm the head of our data and tech policy and public affairs here at the BIA. And I'm just going to give a brief introduction to some of the work that the BIA does in this area before handing over to our panelists today. So as the trade body for the innovative life science sector, we're really aware about the importance of animals in research. I just wanted to outline some of the engagement we have in this space. We're part of Understanding Animal Research, which is a UK not-for-profit organisation that focuses on public engagement and accurate information about animals in research. And as part of this, we're signatories of the Concordat on Openness on Animal Research in the UK, along with about 130 other UK organisations. We're also part of the UK Bioscience Sector Coalition, which is a group of organisations across the UK with an interest in animals in research, and it brings members' views together to inform policy and consultations. And alongside and through these groups, we engage closely with the Home Office via the Animals in Science Policy Regulation Unit. But we're also acutely aware that the UK is a leader in the 3 Rs, and we're also a champion for the 3 Rs in science. And I just wanted to nod to some work that we've done in the past few years to support this in the space. We inputted into the Parliamentary Office of Science and Technology's research briefing on technology alternatives to animals and life sciences last year. We also fed into the development of the Life Science Sector Plan last year, which features commitments on a preclinical translational infrastructure to drive the development of preclinical models. And there was also a commitment on publishing a Replacing Animals in Science strategy, which was published last year, and we were also consulted on. And I think you'll hear more about those later on in the webinar. And I also wanted to nod to the fact that we do a lot of work in the tech bio space and a big champion of the use of technology and AI in life sciences, and a real advocate of the fact that that has really big potential to support the 3 Rs. And if you want to find out more about our work in tech bio, then you can visit our report via the link there. So that's it from me, and now I'm going to introduce today's panelists. We're really excited today to host Daniela Conacchia, Director of Advanced Cell Model Innovation in Safety Sciences at AstraZeneca. We have Anthony Holmes, who is Director of Science and Technology at the NC3Rs, and Thomas Kozdrasky, who is Chief Scientific Officer of CN Bio. So we're going to have short presentations from each of these, and there'll be time for questions at the end. So please do submit your questions anytime throughout the talk via the questions option, the questions feature in the webinar, and then we'll be able to field those to the panelists at the end of the session. So without further ado, I'm going to hand over to Anthony. Over to you.
Thanks very much, Emma. And thank you for a kind introduction, and thank you to the BIA for hosting this webinar on this really timely topic. So good afternoon, everybody. My intention today is really to try and set the scene for the speakers that follow by providing a high-level overview of some of the global regulatory and policy changes that are helping to really drive this transition towards greater acceptance of these more human relevant, potentially more translatable methods that have the potential to replace animals. Next slide, please, Emma. So I do believe that we're at a tipping point where we're seeing real critical mass converging around these methods that's really contributing to accelerating their pace of acceptance. I love this quote from Max Planck. It really does support that idea of the need for critical mass to drive the adoption of innovative practices within science. I think it's often summarized as being a science progresses one funeral at a time kind of approach, suggesting that established scientists can potentially be resistant to new ideas, and that progress comes from sort of a new generation of researchers that accept new opportunities more readily, perhaps. But I think what we're seeing in this space, and what I hope to kind of convey today before hearing from our two other speakers, is that I think something different is happening here in this particular space. I don't think we're in this situation where we're waiting for the naysayers to fade away. And I really do think that we're starting to see critical mass being built actively through a convergence of regulatory and political commitment, technological advances, and scientific capabilities, all driving the acceptance of these approaches to really maximize and realize the scientific and animal welfare, economic and societal benefits that they can bring. And so what I'm going to do today is to focus on more of the regulatory and political drivers for change, and I'm going to leave the scientific and technical advances to both Tom and Daniela, who are after me, who are far better qualified than I am to cover these. And the focus for me will be mostly on that kind of farmer space as well. So next slide, please, Emma. So I'm going to start with a look at Europe and specifically the European Medicines Agency, who I think are developing some really leading best practice and advancing the use of non-animal technologies and methods for regulatory decision making. I think this is really important because the regulators are often seen as being overly conservative and potentially blockers to the adoption of new technologies and approaches for regulatory decision making. And you can understand why that's the case. But I really do think this is changing. And so the EMA have this really well-established working party that's focused on the 3Rs and the use of animals in regulatory testing. And they publish a work plan that describes how they want to advance specific 3Rs opportunities in the short, medium, and longer term. And this can include examples around supporting the integration of in silico methods, and also developing regulatory acceptance criteria for non-animal methods with specific context of use, for example, heart and liver chips for use in organ-specific toxicity testing. Next slide, please. They also have, I think, fairly uniquely specific guidance on the regulatory acceptance of new approach methodologies, and they've also established several mechanisms for engagement on this based on whether you are a test method developer or whether you are a pharma company or a drug developer looking to apply these different methods. And this might seem like a fairly small thing, but I think having this external facing statement that the EMA really is open for business on these technologies is very important in supporting pharma companies and method developers to share their experiences in using these models, but also to support that greater awareness and education within the regulatory assessors around the types of models that exist, how they're applied, and how that data that's generated within them can be used for making decisions. And the EMA have done an analysis of how these different mechanisms are being used, which was published last year, which really highlighted that there's been an increase in engagement in the application or in discussions that are looking to apply these novel alternative methods with regulatory assessors and with the EMA. So as a mechanism, it's working to really foster those communications and discussions that are so important in facilitating the uptake of these technologies. Next slide, please, Emma. So looking now at the pharma industry, specifically the European Federation of Pharmaceutical Industries and Associations, or FPIA. So FPIA as a body and its members really are committed to phasing in the use of non-animal based approaches, and they have a published strategy on how they intend to do this. And they've adopted a unique part of this is the adoption of a 3 baskets approach to group different animal tests. So basket 1 are animal experiments where alternative technologies exist and are ready to be used. Basket 2 are those animal tests where an alternative approach exists but perhaps requires further development or validation, or where a test doesn't exist but there is a clear understanding of what's required to develop an alternative to that particular test. And basket 3 are those animal tests where no alternative currently exists. And this kind of approach really does provide FPIA and its members with a way of streamlining industry's engagement with regulators around this issue to try and accelerate the phase out of obsolete tests and to really focus investment and collaborative efforts where they're most needed. Next slide, please. And lastly, in this kind of whistle-stop tour of Europe, the European Commission later this year, in fact, by the end of Q1, so I think they're looking into the latest timeline is the end of March, to publish their own roadmap on supporting the phasing out of animal testing for chemical safety assessments. So something to look out for there as well. Next slide, please, Emma. So leaving Europe behind and moving over to the US, I think it's fair to say that there really has been limited coordinated federal activity on the 3Rs in the US until about 1997 when they created ICVAM, which is their Center for the Validation of All Alternative Methods, the focus of which is to really try and accelerate that regulatory acceptance of alternative methods. And then a decade later, the National Research Councils published their toxicity testing in the 21st century report, which really provided a significant call for a shift toward human relevant assays and computational models. And since then, we've seen a number of initiatives come out of both the FDA and the National Institutes of Health to really advance this field at a steady pace. So the FDA, for example, have made a very strong signal towards growing openness on the application of tools and technologies such as organs on chips and AI systems through the establishment of their alternative methods working group. The ISTAND program, which is a way of bringing innovative new tools and technologies into regulatory science, and through the Modernisation Act, the FDA Modernisation Act, which eliminates the federal requirement for animal testing for new drug approvals. In 2024, we saw the NIH launch a new program of work called Complementary with $400 million and to be run over 10 years. And this program is really to try and accelerate the development and validation of uptake of human-based non-animal methods. And it includes within it the capacity to establish technology development centers, to establish a validation network, and also to establish a number of shared data resources as well to drive their ambitions forward. And then last year, we saw the FDA publish their roadmap to phase out animal safety testing within 3 to 5 years. And we also saw a number of announcements coming out of the NIH, which have really refocused their funding towards non-animal methods. They've established a new Office for Research, Innovation, Validation and Application, or ARIVA, which will coordinate NIH-wide efforts to develop and scale the use of non-animal approaches. And finally, towards the end of last year, they also launched, with $80 million, Standardized Organoid Modeling Center dedicated to developing standardized organoid-based methods. And you can be sure that in the next, over the next few months, I think further announcements will come out of the US across a number of different agencies looking to advance those ambitions as some of those become a little bit more established. Next slide, please. And then moving away a little bit from the pharma side of things, so just a couple of weeks ago, we saw that the US Environmental Protection Agency are re-establishing their commitment to phase out all animal testing by 2035. So this was a deadline that was originally announced back in 2019, but it was cancelled in 2024 under the previous administration because it was considered to be too ambitious and not sufficiently grounded in science to be achievable. However, under the current administration, they've reassessed that and they've recommitted to this ambitious goal. And so I think, there'll be some really exciting opportunities coming out of that program of work that could be applicable more widely across the sector as well. Next slide, please. So now moving back to the UK, I think it's fair to say that we have been considered to really lead the way in the space of the 3Rs globally, but that has perhaps changed slightly, or that perception may have changed slightly given the extent of activities going on globally and the extent of investment being made internationally in this space. But I can't really talk about a UK perspective without talking about the NC3Rs. And over the last 20 years, as an organisation, we've invested over £60 million in human-based complex cell models. But more than that, we really have helped to build a life sciences ecosystem that's ready and well placed and poised to capitalise on these approaches and to really be in a position where a national strategy, which I'll come to, is achievable within the UK. Next, please, Emma. So alongside the work of the NC3Rs, we're also seeing more targeted initiatives emerging, building capacity in specific non-animal technologies. So this includes the Queen Mary University Centre for Predictive In Vitro Models and the UK Centre of Excellence for In Silico Regulatory Science and Innovation, or CERSI. Next. At a political level, following significant lobbying and by animal rights organisations and increasing animal protest activity, in 2024, the Labour Manifesto committed to working with scientists, industry and civil society to phase out animal testing. Next, please, Emma. And then we really saw this ambition begin to take shape last summer through the publication of the Life Sciences Sector Plan that Emma's already alluded to, which announced up to £30 million for new preclinical infrastructure, but also announced a commitment to a new alternative method strategy to really drive the development and the validation and uptake of non-animal methods in the UK. Next slide, please. And that brings us to this point where, again, as highlighted by Emma, in November last year, our national Replacing Animals in Science strategy was published by Lord Vallance. And this was developed in consultation with government departments, with regulators, industry, academia, funders, and animal protection groups. And the focus really is on animal use across the spectrum of the life sciences and driving change across the whole of the UK life science ecosystem so that we can replace animals with alternative methods wherever possible. Next slide, please, Emma. And to deliver this, the strategy really does focus on 5 key themes that you can see listed here. And under each of these themes, there are several commitments that will have been developed to really deliver lasting change. And each of these commitments have a timeline attached to them, and they also have a designated owner responsible for delivery attached to them as well. So Minister Vance is very keen that this is a very active strategy and that will be implemented at pace with distinct ownership. So I pulled out some of the big ticket items here, but I really would encourage you, if you haven't done so already, to take a look at the strategy and the full list of commitments so you can start to consider how you could potentially participate in delivery of some of these. You can use the QR code below on this slide, sorry, to access that. But I've pulled out some of the big ticket items, if you like, that have been included within the strategy to try and help move this forward. So within discovery research, the strategy commits to establishing a preclinical translational models hub that will bring together different disciplines to generate new collaborations in key health research areas and really try and create and build confidence in non-animal models in sort of the disease modeling and efficacy setting. Within regulatory decision making, we have said that we would establish a UK centre for the validation of alternative methods that will focus on validating models to accelerate both industry and regulatory confidence and acceptance for regulatory decision making, particularly in the areas of safety and toxicity testing. Unsurprisingly, there's a big focus on data, and one of the commitments within that is around trying to generate and support the development of data sharing platforms that will really maximize the accessibility of public and private data sources for the development and training of predictive models. We want to be seen as supporting and leading internationally in this space. We've committed to hosting a meeting of regulators from across sectors globally to try and really agree actions that will incentivize and speed up the validation and acceptance of alternative methods and really support our efforts or the international efforts for harmonization around some of these methods and their adoption. And then finally, there's a really strong focus around governance and delivery. So delivery of the strategy will be under the ministerial leadership with a cross-government ministerial committee that's led by Minister Vance. And there will also be a publicly available and facing dashboard of progress against key deliverables for full transparency. Next slide, please, Emma. And we're already seeing that delivery of the strategy is taking place and it will continue to take place at pace. Like I said, Minister Vance is very keen that this moves quickly and that we deliver on the strategy in the way that we said we will do. So at the end of last year, we've already seen that the MRC, Wellcome Trust and Innovate UK, working in partnership with the NC3Rs, has committed nearly £16 million to support the development of human-based in vitro models of disease. And we've also seen that next month, the NC3Rs and Innovate UK are bringing together stakeholders around reducing the use of animals in pharmacokinetic and cardiovascular safety studies, which are 2 areas that have been specifically described within the strategy as priorities for new model development. So registration for that workshop is open until the 27th of February. And again, you can access the information there through the QR code on screen. So if you have some interest in this space or you feel that you've got some technology development that can support this, please do register for that. Next slide, please, Emma. And so you recall at the start of the presentation, I showed you this quote from Max Planck on his view of how or what's required for the acceptance of new scientific approaches. Next slide, please. But I think, what's happening in this space is that the non-animal methods community, and I'm including all stakeholders in this, it's really being driven by a critical mass of leaders who together are really building this culture that embraces innovation and which is driving that global shift in acceptance. Next slide, please, Emma. With that, I'd like to just thank you for your attention and very happy to continue the discussion during the panel session. But it's my pleasure now to hand over to the first of our 2 remaining speakers, Dr. Tom Kozichevsky from CM Bio. Tom, over to you.
Thanks, Anne. Good afternoon, everybody. Great to be here. Thanks to the BIA for the opportunity to talk to you all. So I'm going to follow on from Anne's great introduction and show you a little bit more about what these new alternative methods are and how they're being used and adopted, with a kind of particular focus on the pharma biotech industry. So next slide, Emma. So high level intro, drug discovery is hard, and it's getting harder. Our ability to get new drugs to patients is getting ever tougher. And so the real focus needs to be on improving the way we do preclinical medicine and preclinical discovery. And we really need to do new methods to do that. We need that for a whole bunch of different reasons, as well as the drive to reduce animal use. There's this ever broader swathe of new modalities being developed that are human specific. And so we really need preclinical models that are human focused around human biology. They're agnostic to the different modalities that are being developed against different targets. They capture complexities of human biology, such as immunology, and they're usable and accessible, and that's really important for scientists to adopt these. So next slide, Emma. So human biology is infinitely complicated, and I think we're finding it more complicated all the time. And when we build these types of technologies, such like we do at CM Bio, we've really got to think about how much of this complexity of human biology we can capture. So if we're modeling a disease, is it an acute or is it a chronic disease? What are the drivers of that disease? And how many of those things can we recapitulate? On the flip side, there's a lot of use of these tools to assess toxicity. So what are those toxicity mechanisms actually observed in patients, and can we really truly recapitulate those? Is it being driven by an immunological response, or is it a direct effect on a particular tissue? Does it affect a certain population, for example? So next slide, Emma. So the way that we think about this, and that all these new tools, which are fantastic, are really in a toolbox for a preclinical researcher. So there are those traditional tools, growing cells in two-dimensional flat dishes, using in vivo models, and then all these other new alternative methods, NAMs, come alongside them. And all of these tools have their utility and their pros and their cons. So I'm going to give you an overview of them today and walk you through some of the major ones. So next slide. So first of all, in silico, computational tools are obviously rife across the whole of society now, but particularly they're being used broadly across drug discovery in a number of different areas. So starting from the way we identify and validate targets, the way that we design drug molecules and direct these against those targets. These can all be done computationally now. We can filter down the number of these lead molecules that we have, all computationally based on the data sets that we have. And then in later stage of preclinical development, in silico models are being used to predict exposure profiles, so ADME profiles of how these drugs will expose to human tissues in the body. But what's fundamental to all of these tools is they have to be fed by data, and they have to be fed by quality data. And so there is a real need for better quality data to feed these models, not data that's necessarily generated from an animal model that might not be predictive. And certainly clinical data is obviously patchy at best some of the time. And I'll come back to this later, but the combination of in silico approaches and AI with other in vitro approaches is going to be fundamental to the future adoption of these types of technologies. So next slide, Emma. So the first kind of major area of in vitro NAMs is the spheroid and organoid field. So spheroids have been around quite a long time and now quite widely adopted. These are three-dimensional, ultimately balls of cells. They can be a single cell type or multiple cell types. These are brought together mainly in ultra-low adhesion plates. And these can be relatively simple or more complex depending on the model that you want to build. They have great benefits. They're relatively simple and relatively cheap to pull together, but some of the complexity can be lacking in certain applications. A number of years ago, researchers in Europe developed and have now really scaled the organoid technology. And again, this has now become much more commonplace amongst research institutes. Now, organoids are derived from human tissues in some way, either from tissue fragments or from isolated stem cells that are then specifically differentiated, under specific conditions. So specific media, cytokines, growth factors, and that these can then be matured into many different microtissue-like structures, organoid structures and there are huge numbers of papers now been published on the generation of organoids for all sorts of different human tissues, and they form these globular-like, very small structures that can be grown in dishes in very specific matrices, very specific conditions. And they start to mimic tissue and organ level function. And so these are a step above what can be done in a spheroid. And again, these can be handled in potentially high throughput plates, which can bring a number of benefits for scaling. But again, these models do have some challenges with the levels of complexity that they can generate and some of the maturity of some of the tissues that they can generate. So there's a really great place for these types of models, but they can also be supplemented by even more advanced in vitro models. So next slide, Emma. So one area that's been around for a long time is the use of actual human tissues. So these have typically come from patients, from surgical resections, where little pieces of human tissues are sliced and prepared and then put into the lab in cell culture conditions. And these can be great. They maintain all the histological features of a human tissue. But typically when you put these in the lab, they only last for a couple of days. So they can be very useful to answer very specific questions. But typically over time, some of that tissue starts to degrade because they're no longer in the patient's body. One new advance, and you sort of see an image on the bottom left-hand side here, is the ability to bioprint whole human tissues now. And this is a growing area in the NAM space, and these things will start to combine in the future years. This is still a relatively new area of NAM technology, but again, an area of particular excitement. So next slide, Emma. And then the other major arm of the NAM space is microphysiological systems, MPS, or also known more colloquially as organs on chip. And these are microfluidic bioengineered devices for growing cells. And they typically are used to grow human cells and deal with the challenge that growing human cells, or particularly primary human cells in vitro, is incredibly hard and finicky. And these models create the right microenvironment to grow these cells as optimally as possible. And they control as many different features of that microenvironment as possible. And they typically use microfluidics to allow a perfusion of cell culture media within these devices to mimic blood flow. They can use other features to allow tissues to flex or grow. So, for example, cardiac tissues can pulse. And they can control much more accurately the way that drugs or hormones or other key factors then interface with these mini microtissues. And more advanced than that, there are now a number of multi-organ MPS systems that are available where we can actually link multiple organs together to ask even more complex questions in vitro. So, for example, you can link together, say, the intestine and the liver to look at the absorption of an orally given drug and how it then goes and distributes around the human body. Next slide, please. And a whole plethora of MPS technologies have been developed by the community over the last sort of 15 to 20 years. And they're growing in complexity all the time, but they're also growing in utility and usability. And these can be used to model disease states, but they can also be used to model toxicity and those ADME exposure profiles that I referred to earlier. And it's this kind of data that they can feed back into those AI models. And other key areas of being focused on are such as the incorporation of immune components that are so fundamental to disease and toxicity, but also things like the microbiome and other areas are being built into these models as they're becoming more complex and widespread. Next slide, please. So a typical use of one of these systems, they can take a plethora of different cells to start with that are seeded in or added into to one of these devices. And what's probably different to a lot of other approaches is they are typically then cultured or grown for a number of weeks rather than a number of days. And this starts to mimic in vivo biology much more closely. And you can mimic things like chronic dosing of a drug or an induction of a disease state that occurs over many weeks rather than just a number of days. And then a whole slew of analytics that have been developed over many years, high-level microscopy, omics, metabolomics, transcriptomics, and particularly looking at histology and quantitative translational endpoints is key from any of these in vitro models to take the data and translate it at the end from what comes out of one of these advanced in vitro platforms to predict what's going to happen in the clinic. Next slide, Em. And so these tools are now being used throughout drug discovery by different companies at different rates and at different stages. So some of those higher throughput spheroid and type organoid models I referred to being used in maybe early target discovery, some of the more advanced complex models in that lead optimization phase. And in that regulated phase, you can see all the things that Ant talked about. These tools are now starting to be qualified and validated for that late stage preclinical development. And they're also being used to support clinical development, where companies are having challenges in the clinic, and they need to unpick the mechanisms of what they're seeing in their patients, and then they're coming back and using technologies, for example, like the MPS technologies we have at CMBio, to really unpick the questions that researchers are having clinically, and to understand why they might be seeing certain things and taking those forward. Next slide, Em. So I'm just going to give you a couple of real world examples of how some different NAMs are actually being used to influence regulatory decisions. So a really exciting field is the iPS derived neuronal field, where there's some incredible work done to build motor neuron models, neurospheres, and these are now being used to model human brain diseases far more accurately than has ever been done in vivo. The model I show that on an image here is of a microglia, which is key to model human ALS. On the top right of this slide, you see images of a skin sensitization model. These are used to model allergic skin responses. These are now very well established, and there are very well established guidelines for using these models to assess skin reaction and skin sensitization. Those digital models, such as QPS modeling, are now being used extensively, and there's some really nice examples of how these can be used to optimize dosing strategies in the clinic and support clinical development of, for example, bispecific antibodies. In the MPS space, there are a number of examples of how MPS models have been used to drive clinical and support IND filings. And for example, at CMBio, we've developed a very advanced model of human liver disease and metabolic liver disease, and we've used that to help a number of clients move their molecules into the clinic and go and treat patients for these diseases. Next slide. So I've alluded to already, I think one of the really key pieces for the future is bringing these tools together, not using them in isolation, and in particular by combining together the advances in these in vitro MPS models with these PBPK models, AI and preclinical models. We'll be able to really take the data we generate to the next level. And you can imagine a world that's not too far away where we can take a range of different MPS type endpoints, and we can use computational models to bring them together and then ultimately predict whether a drug is going to be safe, how it's going to be exposed in a human to all the different tissues of the body, and get those readouts. Not all of those tools are available now, but they're all being built, and AI and computational model will be like the glue to bring them together. And again, by supplementing that with multi-organ MPS, they'll become a really powerful data set. Next slide, Em. So to support this and how we're going to drive this further, well, what's important is for each of these tools, you've really got to focus on the problem it's trying to solve. What's the context of use where it's really going to be most effective? Not just simply building the greatest model of the human liver or the human lung. There are lots of activities, and referred to some of them already, and there's more looking at how do you define validation and performance metrics for these types of models, and defining the right benchmarks for showing that they are fit for purpose. And one thing that's really going to drive this is collaboration and working together cross-functionally to really drive the adoption of these models across the industry. Next slide. And just as some examples in the MPS space, there are a number of international groups now working incredibly hard. The International MPS Society was established a number of years ago. It has now become a real cornerstone of the industry. The IQ MPS affiliate is a fantastic group of pre-competitive pharma companies coming together to share their insights in using these types of technology and how they drive them forward. Next slide, Emma. And so to give you an example of one area that we've been working on, led by the 3Rs Collaborative in the US and CPATH and the NIH, is we've been working with a group of other commercial developers of liver MPS technologies to demonstrate how our platforms can be used for drug-induced liver injury and support when this incidence and this toxicity is observed in clinical trials. We've been working together, working with FDA, and this project has been accepted into the iStand qualification program that Ant referred to earlier. And so we have a consistent protocol for running these 8 different liver platforms in parallel. We've been screening it, for example, with blinded drugs to demonstrate how it can be qualified and validated. And more data from this is going to be published throughout the rest of this year at a number of key events, and look out for that. Next slide, please, Emma. So to sort of round off, I think that the future for NAMs is very exciting, but there's a lot more work to do. There's a lot of mountain to climb, and there's plenty of challenges for all of us to try and solve. So next slide, Emma. So just a couple of these to finish. So I already touched on that integration piece, and that's going to be key. Bringing together multiple tools, not one tool or one approach is going to be suited to fix and solve everything. Modeling systemic responses is still going to be the biggest ultimate challenge, and that will be probably the most hardest thing to come through. The development of immunological models still remains a challenge. The human immune system is just an incredibly powerful thing, which we're only just really starting to understand, and modeling that in these models is still not straightforward, but is being developed all the time. And all of these in vitro models do need consistent sources of cells, high quality, very highly phenotypically relevant cells. There are lots of cells out there, and speaking from personal experience, they're not all made equal. And so high quality cells that we can rely on that supply chain is going to be fundamental to our future success. And data, having comparison data from clinic or other approaches is going to be fundamental to allow us to be successful and really drive the adoption of these technologies into the future. Next slide. So just as a final thought, these technologies are really here now. They're here for the present, and a number of them are very much fit for purpose and ready to use now, but they can't solve every problem. So there's a lot more work to do, and that's got to be led by that rigorous science that underpins everything. Collaborative activities pre-competitively and across parties is going to be fundamental so that all the stakeholders are engaged and brought along for the journey and can guide and shape where we go. So next slide, Emma. And with that, I'll say thank you for listening. If you're interested to learn more about CM Bio, then please visit our website and look forward to the discussion and later. And I'll now hand over to Daniela from AstraZeneca to show you how they are using NAMs in their work.
Thank you very much, Tom, for a really nice introduction and a beautiful seminar. And also thanks to the organizers today for inviting me to participate in this webinar. So today I would like to share with you how we at AstraZeneca use advanced cell models and new approach methodologies to make better predictions for drug safety. Predictions that are not only more relevant for humans, but also can help us reduce our reliance on animal models. Next slide, please. So I'll start by giving you a brief overview of AstraZeneca. Here we are a global biopharmaceutical company with some key therapy areas, one being oncology, and then biopharmaceuticals, which covers cardiovascular, renal, and metabolism, respiratory and immunology, and vaccines, as well as rare disease. And in all of these therapy areas, our goal is to create novel cures for patients that are afflicted by serious and chronic conditions. Next slide, please. So in the past years, the way we create medicines has evolved tremendously. From drugs being mostly based on small molecules, now we are increasingly developing our medicines using large complex molecules and sometimes even cells. And these are drugs such as antibodies, antibody drug conjugates, and antisense nucleotides. And these are collectively known as new therapeutic modalities or biologics. And what distinguishes these is their exquisite specificity for their target, in this case, the human target, which of course creates a transformative opportunity to cure human disease, but at the same time, it also creates novel challenges in how to profile the safety and efficacy of these new medicinal modalities. Next slide, please. And this is why today, more than ever, new approaches are needed to create safe and efficacious medicines. Traditionally, the preclinical drug development process relied on two main types of models, so two main types of tools, simple cell models and animal models. Simple cell cultures, which are often derived from cancer cells, are not physiologically relevant replicas of human tissue. They do not reproduce cell type fidelity, complexity, and functionality. And on the other hand, animal models, which do provide the complexity and insight into systemic drug effects, animals are not humans, and they are becoming increasingly less reliable to predict patient outcomes, especially when testing new modalities. And this is where advanced cell models come into play. These are human cell-based systems that offer increased complexity and also cell type fidelity. They can be derived from stem cells, as Tom has really nice described, but also from primary tissue. They can be grown in 2D, but also be three-dimensionally organized, such as organized and spheroids. They can be of increased complexity with the immune component being integrated, as well as be incorporated into microphysiological systems where they're exposed to microfluidic perfusion and allow us to mimic multi-organ crosstalk. Next slide, please. And at AstraZeneca, we have developed a battery of different advanced cell models that cover the tissues and organ systems that are mostly affected by drug toxicity. And we also integrate immune cells when relevant and when necessary to model the effect on the immune system, for instance, for immune modulatory drugs. But because advanced cell models do not alone tell us exactly what's going to happen in the patient, we combine these in vitro approaches with in silico methods, such as mathematical modeling, like quantitative systems toxicology or pharmacology, known as QST, which integrates advanced data from advanced cell models, available clinical data, and also known PK/PD data to model more accurately the drug exposure and predict patient responses. So this integrated approach allows us to make faster, more accurate decisions, reduce the reliance on animal models, as well as increase our chances of clinical success. Next slide, please. And this is not just theoretical. We are applying this integrated approach across our whole drug development portfolio, from small molecules to large complex biologicals. And we have seen a surge in the number of AZ projects that are supported by advanced cell models just in the last few years. Next slide, please. But now I would like to move on to give you a few specific examples of how this works in practice. For instance, by showing you how we leverage microphysiological system of the bone marrow to resolve a small molecule-driven toxicity of the bone marrow. Next slide, please. So it is known that traditional models do not capture accurately toxicity of the bone marrow, which is why we developed internally a model of the human bone marrow relying on the tissues CHIP-2 system. And here in one of the chambers of the CHIP, we place a ceramic scaffold that is modeling or reproducing the human bone, on which we can grow hematopoietic stem cells together with mesenchymal stem cells. And under these conditions, the hematopoietic stem cells can differentiate and give rise to three of the main lineages of the blood, namely erythroid, myeloid, and megakaryocytes. And we can sample the cell viability as well as the effect of drug toxicity on these cells over time by analyzing them through flow cytometry. Next slide, please. And next again. Thank you. And this is an example of a study where we tested a small molecule that was predicted to have bone marrow toxicity in our bone marrow MPS. Traditionally, these assessments would be conducted in rat in vivo models. However, in this case, the project lacked the confidence of the translatability of the rat model to human, which is why in this case, we directly tested our lead compound in the bone marrow MPS. And what we observed was a dose-dependent effect on the bone marrow that predominantly affected the erythroid lineage. And these data recapitulated what had been previously seen in vivo in the rats, which also provided validation for the model. We then fed these data onto our QST mathematical modeling algorithm, and this predicted that there would be no bone marrow toxicity at therapeutic exposure with a threefold margin to mild anemia. And this provided the project with the confidence to progress the molecule. So taken together, this demonstrates the power of coupling advanced cell models and QST modeling to guide decision-making for safety. Next slide, please. Next again, thank you. Yes, so this brings us directly to the impact of using these models, because we have built over time confidence in these models by validating them robustly with in vitro data and in vivo data alongside each other, as well as by integrating clinical data when available. we have demonstrated repeatedly that the bone marrow NPS consistently provides increased accuracy of clinical outcomes, and which is why now over the rat in vivo model. Now, this is the reason why now we routinely recommend that assessment of hematotoxicity, so bone marrow toxicity, is conducted in MPS, in bone marrow MPS, and no longer in rat in vivo studies. And this has, over the past just a few years, led to a complete replacement of the use of animal models with advanced cell models specifically for the hematotoxicity assessment. Next slide, please. But if we move away from small molecules and towards new modalities, so biologics such as antibody-drug conjugates or ADCs. Next slide, please. ADCs are antibody-drug conjugates, and these are antibodies that are conjugated to highly cytotoxic compounds that are designed to target specific antigens on cells, and most often neoantigens on cancer cells. So this is used as a cancer drug. However, the toxicity of ADC is more often linked to either its off-target delivery to non-cancer cells or to the release of the cytotoxic payload by dying cells towards adjacent healthy tissue. So this means that the therapeutic window of ADCs is relatively narrow, which means that the difference between a dose that is efficacious but safe and one that is toxic is very small. This means preclinical safety testing really critical for ADC development. Next slide, please. So what is the main toxicity that is observed upon ADC treatment? It is peripheral neuropathy that is observed in patients and manifests as tingling, numbness, and pain, as well as an impairment of fine motor skills in patients that are treated with ADCs. And this is most often the case for ADCs that are conjugated with microtubule inhibitors such as MMAE. The problem with this type of toxicity is that it cannot be captured in animal models, in vivo studies. In fact, rats and mice that are treated with ADCs at standard doses and durations do not develop the nerve damage that we observe in the patients. And so this is a perfect example of where species differences fail us in predicting patient outcomes, and where human-based models are needed to design safer molecules before they reach the patients. Next slide, please. So in line with this, at AstraZeneca, we assess ADC-induced peripheral neuropathy using an in vitro model of human sensory neurons, which is the cell type that is affected in patients. And when we treat these neurons with ADCs, as you can see on the left side, that are known in the clinic to induce peripheral neuropathy, what we can see is that, in fact, we can detect neurite degeneration in this in vitro model. However, on the right-hand side, if we treat the same model with ADCs that are conjugated to a different cytotoxic agent that is known to not cause peripheral neuropathy in the patient, such as topoisomerase 1 inhibitor, here we see that the neurites are spared. So this indicates that the model is specific, so it can distinguish between neurotoxic ADCs and non-neurotoxic ADCs. And it also indicates, which is reflected in the bar graph at the bottom, that it can detect toxicities at doses that are in a clinically relevant range. And finally, while peripheral neuropathy is developed in patients over weeks or months, with this model, we are able to detect neurodegeneration over just a few days of treatment, which significantly accelerates decision-making. Next slide, please. Next slide, please, again. Okay, so and now I will show you an example of how we use intestinal organoids to model gastrointestinal toxicity, which is a common and sometimes severe side effect of cancer drugs. Next slide, please. Next slide, please. And for this, we use, it's okay. For this, we use intestinal organoids. So intestinal organoids are, they recapitulate the intestinal crypt which is the regenerative and proliferative compartment of the gut and is the tissue that is mostly affected by cancer drugs, which results in clinical diarrhea. And this is an example of how we use intestinal organoids to predict clinical gastrointestinal toxicity. So you can see on the left-hand side that when treating the organoids with drugs, we can detect toxicity by imaging as a result of reduced growth of the organoids. In the middle, when this data is then fed onto our QST mathematical model, it generates clinical predictions, and the results are really impressive. In fact, if you look on the right side, you can see that for 5-FU, which is a standard chemotherapeutic, we predicted with our data from advanced cell models coupled to mathematical modeling that clinical diarrhea would be observed in 35% of cases, whereas the actual observed incidence in the clinic was 22%, so really close to our predictions. But at the bottom side, you can see where this approach becomes really powerful. So drug A is a compound where mice showed no GI toxicity, even at 50 times the equivalent human dose. So based on this mouse data, this drug was supposed to be safe. However, our organoid model told a really different story. In fact, for the low dose, we predicted a 35% diarrhea incidence using the GI organoids, whereas the actual clinical rate was 24 to 58%. For the high dose in yellow, we predicted 60% incidence, whereas the actual rate was 51 to 75% of patients being affected by this condition. So in essence, our human model was right, whereas the mouse was wrong. And again, this demonstrates why human-based models are so valuable, and in the sense that they can catch toxicities that animal models can miss, potentially preventing clinical trial failures and protecting patients from unnecessary side effects. Next slide, please. And I know we're almost over time, we're on time, so I will only give you a glimpse at this point how we integrate advanced cell models with omics and AI to create next generation platforms. And we refer to this integrated safety strategy as AI safety omics. And the goal here is not only to humanize and accelerate the safety cascade, but also to leverage unbiased multidimensional data, such as cell painting and multi-omics, to generate safety predictions with unprecedented depth. And for this, we have already moved beyond the vision, and we're already applying this to the benefit of the portfolio, where this strategy allows us to select safer chemistry much faster. And when we apply this at scale, we're able to identify safer molecules. And traditionally, advanced cell models, the challenge with the advanced cell models has been the cost and the scalability. So our recipe here is miniaturization and automation, which allows us to deploy these technologies at scale and in a cost-effective way. And with this, I would like to conclude. Next slide, please. And thank the incredible team that has made this work possible and that keeps relentlessly pushing the boundaries of science to make better medicines. And thank you for your attention.
Thank you. Thank you so much, Daniela, Tom, and Anthony. These talks are really, really interesting. And I think what I love about this area is like the science and the policy are sort of at this point where they're really reinforcing each other, aren't they? And sort of building momentum. So we don't have heaps of time, but there's been a couple of questions. So I think we have time for those. Let me have a look. So one for Tom. One of you, how implementing the research reflects on the cost of research. So multiple technologies have to be incorporated to answer questions, and how does the cost of the techniques and the technologies compare to animal use?
So that's a very broad question, very individual organization specific. I think that Daniela pointed to it at the end of her talk as well, right? I think some of this comes from scale. I think adopting any new technology in the first instance, has got to take there is an amount of cost involved up front, but there are a number of ways to mitigate that. There are a number of ways to access technology now. Certainly something that we're doing is working with big global CROs. So these technologies are going to be offered through those routes too. So before you bring technologies in-house, to use at scale, we see that automation is a big thing, and that's going in across the industry. I think ultimately that a lot of, particularly in certain academia, There's this reliant on animal houses, right? They're embedded in institutes, and certainly something that we're I know talking to numerous people about is an idea that, well, can't we take some of that space and use it to make NAM based hubs, right, in institutes? And I think that's a really kind of exciting initiative. Use that space, have some core facility with a little bit of expertise to kind of give people a starting point. So I think there's a number of things like that going on.
And then I think another question that crossed my mind when you're talking about data generation, because we did this big feature last year at the BIA on functional genomics and generating more functional genomics data. So I guess, to any of you who wants to take it, where do you think we've got a kind of a particular desert in data that could really support this area?
So I guess I'll jump in first, but then I'll let the others go. Like data is key, right? Data is the most important thing. The model is actually not the important thing, right? The data is the fundamental thing at the end. So from an organization like AstraZeneca, right, they want to look after and preserve their data to some or a lesser extent for fairly obvious reasons. And I think we've got to find creative ways to strike the balance between companies being able to generate their own data sets, hold them, utilize them for their own purposes that they've legitimately developed, but also have an amount of data that the community as a whole can share and grow from. Because obviously the analytical AI tools are there to assess the data. There's just a lot of it is not ready and shareable. And also something that someone said to me recently is that even if we did all make our data shareable and available across every organization, there probably isn't enough anyway. So there is a need to generate more data across all fields, and that's a kind of I know that's a responsibility for a lot of people.
Cool. That makes sense. Thank you. And then, yeah, I think we're probably at time. There was another question about NAMs in neurodegenerative and brain diseases, but I feel that could be a whole subject of a whole talk. I don't know if anyone's got a quick response to that one, or we'll wrap up here.
I think just to say, it is neurodegenerative diseases and sort of brain organoids is an area of real activity at the moment. And, there are a number of reviews out there which are kind of pulling together the latest state of the art and where the gaps are. So I'd encourage whoever asked that question to look at a few of those to get a better understanding of what the landscape looks like.
Thank you. Thanks, Anthony. So finally, just to say, yeah, a big thanks to Anthony, Tom, and Daniela for those talks. Are really, really interesting. It's a really exciting field where things are moving very fast. So hopefully have you back in the future to sort of do an update on all of this. And then just to flag to everyone online that we have a really exciting program events at the BIA. And so do check out our website for more information on in-person and more webinars coming up throughout the year. Thank you, everyone.
Thanks, Emma. Thank you.
Thanks, Emma.
Thanks, everyone.
Thanks, everyone.
Thank you.